The way the genome is folded controls which genes are activated and when. My research group tries to understand how the genome is folded and how its misfolding leads to disease.
My research is at the interface of cell biology and data science. My lab produces biological insight along with specialized analytic tools to reveal hidden patterns within complex microscopy imaging data. We develop and apply a range of methods from computer vision, machine learning, network sciences, and time series analysis.
We study genomics. That means that we read the DNA, the code of life, and we use AI tools in order to interpret what we read and eventually to apply it in the clinic.
We measure experimentally the folding of the genome and use artificial intelligence to try to understand what the rules are for organizing the genome. Deciphering how the genome is folded and what the biological consequences are is going to allow us to better understand disease and potentially improve treatment and diagnosis.
We are generating sequences of synthetic images where we amplify subtle image properties that are critical for the model prediction. We are applying machine learning to in vitro fertilization data. Also, in an international consortium, we're integrating different imaging modalities to predict and characterize cancer tissue disease states.
In one of our studies, we were able to read the entire tiny pieces of DNA in a mother's blood and to separate them into embryonic DNA and mother's DNA. Now we can focus on the embryo, the future baby, and identify point mutations that could be detrimental later on in life.
The Technion Faculty of Medicine provides a special interdisciplinary environment which is really critical for this problem. My research team consists of students from math, computer science, biology, and chemistry coming together to solve this problem.
Our research is made possible thanks to a fantastic IT staff at our Department of Software and Information Systems Engineering at Ben-Gurion University.
In our lab, we are a hub of various fields. We combine them all together and we use AI tools in order to reach better decisions next to the bedside.
We aim to use our discoveries to build new predictive models that will allow us to translate the language of DNA into genome structure and biological function.
Tel Aviv University is an extremely innovative and exciting place to do AI. We collaborate among departments and schools, and we drive our questions forward in a collaborative way.
The long-term goal of my research program is contributing data-driven modeling to generate mechanistic insight of complex and dynamic cellular functions in health and disease.
When we bring our insights into the clinic, the clinical team can take better decisions. This will result eventually in not only extending life, but also improving quality of life.